So let's move on to the second talk session of ACCSA. I would like to introduce Dr. Pragati Priyadarshini Balasubramaniam from IIT Kanpur. Dr. Pragati is an assistant professor in Cognitive Science Department at Indian Institute of Technology, Kanpur. She directs Transitional Neuroscience and Technology Transit Lab focused on the science of cognitive brain computer interfaces and developing technologies for clinical applications or interventions at IIT Kanpur. Much of her research work can be categorized under cognitive, computational, and transitional neuroscience. She completed her PhD from Indian Institute of Technology, Madras, India, and did her postdoctoral research with Professor Benjamin Hayden at University of Rochester. She has worked as an assistant professor scientist at University of California, San Diego in the Neural Engineering and Transitional Labs. She co-founded an initiative to translate popular science to local languages called Project Kaleidoscope and made science available at local libraries in Tamil Nadu. I heartily welcome Dr. Pragati to chair the second talk session of ACCSA. I would also like to invite Mrs. Arathi Rajendran of Amrita Mind Brain Center to co-chair the session.
Thanks a lot, Akhil. That was a very kind introduction. I'm very excited for this talk session too, like all of you. And thanks a lot, Arati, for co-chairing the session with me. So we have a lot of talks, about 9 presentations that we have to cover in about 2 hours. And all of them range from cognitive neural basis to translation. They're all very exciting. We will have pre-recorded presentations just for convenience sake due to COVID situation mostly. They are all about 10 minutes in time. Once the presentation is played, we will have time for a question or two. I'll please do direct your questions to me in the chat box. I'll try my best to ask as many questions as possible. If you are not able to get to them, please feel free to ask through chat your questions directly to the speaker. Without any further delay, let me introduce the first speaker of the day. We have Dr. Sai Sailesh from R.D. Gardi Medical College, MP, and the presentation is going to be on electrical vestibular nerve stimulation as an alternative therapy to improve spatial and verbal memory in patients with Parkinson's disease. Over to you. Thank you.
Good morning, everyone. I am Dr. Sai Sailesh Kumar from R.D. Gardi Medical College. And title of my presentation is Electrical Vestibular Nerve Stimulation, an alternative therapy to improve spatial and verbal memory in patients with Parkinson's disease. And the study was funded by Indian Council of Medical Research. And the devices, whatever we used, was given by Neurovalence from UK. Parkinson's disease is the second most progressive neurodegenerative disorder with 10 million people affected worldwide. There exists plentiful literature regarding the motor functions of Parkinson's disease, but the literature on non-motor symptoms, especially cognitive functions, is very little. Optimal vestibular stimulation was proven to improve both spatial and verbal memory in healthy individuals. So why this study was undertaken? Because there is no approved treatment for limiting the progression of cognitive decline in Parkinson's patients till date. Hence, the present study was undertaken to observe effectiveness of electrical vestibular nerve stimulation in improving spatial and verbal memory in the patient with Parkinson's disease. So this is a randomized controlled trial, which was registered with clinicaltrial.gov. Participants were assessed at 3 points of time, that is the baseline values, after 6 weeks and after 12 weeks of the intervention. Electrical vestibular nerve stimulation was given as an intervention to the individuals of intervention group, whereas placebo stimulation was given to the individuals of control group. And electrical vestibular nerve stimulation, the bilateral stimulation was administered using battery-powered vestibular nerve stimulator. The total duration of the intervention is 12 weeks with 5 sessions per week. Each session duration is 1 hour, and the placebo stimulation was administered to the control group. The device consists of a headset electrode pad, and skin swabs. The device can be turned on using the power button, and the intensity of the stimulation can be adjusted. So the device will deliver the stimulation level from 1 to 10, and the participants will have opportunity to adjust the level of the stimulation as per their comfort. Majority of these participants used 3 and 4 levels of the stimulation. So that is how exactly the image of the device, what you can able to see. And the right side, there is a app which is available from the Google Play Store. Can download the app and we can connect it to the device. The device can be managed both manually as well as by using the app also. An advantage of using the app is we can able to see how much time we have used the device and other things. Also from the server, we can also see whether really the participants are using it or not, what they are doing, all those things. So study setting is the Department of Physiology in collaboration with the Department of General Medicine of Adigadi Medical College, Ujjain, Madhya Pradesh, India. 30 cases of Parkinson disease patients, including both males and females, were recruited in the study by convenient sampling after obtaining voluntary written informed consent. Patients were recruited from the outpatient ward of General Medicine Department, Adigadi Medical College, and they were randomly assigned to control and intervention groups by random numbers generated through randomizer.org. Inclusion criteria: 18 years and older and willing male and female participants, and stage 1 and 2 of Horn and Yahr classification of disability, ambulate with or without an assistive device for at least 50 feet, were able to get up and down from the floor with minimal assistance or less, and score of 24 or above on the Falstein mini mental state examination. So whoever is fulfilling all these criteria were included in the study. And participants with any severe complications and stage 3 and 4 and who are not willing, all those were excluded from the study. So this consort chart will explain. We have totally assessed 169 participants in the study. Out of that, we excluded 135, and because they are not meeting the criteria, and 34 were there who are randomly assigned into control and intervention group. In the intervention, we have 16, and in the control group, we are 18. But from the control, again, we lost one person because of the location change, and 2 persons reported that they are feeling dizziness. So this is actually placebo group, but they are reporting dizziness, so they discontinued. And again, from the intervention group also, we lost 1 person because of location change. So we ended up with the 15 and 15 in both the arms. So to assess the spatial memory and verbal memory, we prepared a simple charts, which consist of 10 simple images or 10 simple words. So because they're elderly people, they cannot remember very tough words. If we use, they cannot remember. So very simple words, like simple pictures like dog, flowers, trees, like that we put, and we showed them for some. We showed them for 1 minute. After 1 minute, we asked them what is some simple question. What is your name? Just to deviate them. What is your name? What is the day? Like that. After 1 minute, we gave them 1 minute time to recall how many images or how many words they have seen. So correct answer will be given 1 and incorrect answer will be given 0. So same thing, in spite of the image, we have here the words. So simple words, again, very simple words, what they can able to recall easily. So 1 minute we have shown, then 1 minute we deviated them. After that 1 minute, they have to recall whatever they have seen. One example of what we have used. These things were used during the baseline values, and different charts were used at different points of time. Means baseline charts are different, 6 weeks charts is different, 12-week charts is different. The study protocol was approved by Institutional Human Ethical Committee of Adigadi Medical College. Data was analyzed using SPSS 20.0. Two-way ANOVA was applied to observe the significance of difference. Born-Fereni post-test were applied to observe the difference between the groups. Demographic data like age, height, weight, BMI were compared using an unpaired t-test, and a probability value less than 0.05 was considered significant. So demographic data, age, weight, height, BMI was not significantly different between control and intervention groups. There is a significant improvement in the spatial memory as well as verbal memory, both after 6 weeks and after 12 weeks in the intervention group when compared to the control group. So vestibular stimulation improves memory through its connection with the cortical and subcortical structure. This is true because a lesion to vestibular system causes declined memory and reaction time. Vestibular stimulation was reported to modulate the activity of the place cells in the hippocampus. In fact, the lesion of the vestibular system causes atrophy of the hippocampus. So that much connection is there between vestibular system and hippocampus. Vestibular stimulation was also reported to improve attention in the patient with a head injury. So there was a significant improvement in both spatial and verbal memory in the patients after the stimulation. So the study recommends further detailed studies with higher sample size so that we can recommend electrical vestibular nerve stimulation as an adjunctive therapy, and it is a non-invasive and very simple to administer also. And there is no side effects. What we have observed, we have conducted a follow-up study also. There is no side effects reported so far by any patient. So we can recommend that in the management of cognitive decline in patient with Parkinson's disease. These are the references, few references, all references I cannot put. So only the key references I have given here. These are my team who supported me a lot to do this study. Though the study is halfway, because total sample size is 60, we have to go one more year. Thank you to my team. Thank you so much. Thank you for giving the opportunity.
Thank you. That was a wonderful presentation. Thanks a lot. The work is so interesting. Any questions from the audience? Please do write it out in the chat box to me. I haven't received any questions till now, so but I'm excited to ask one question.
Yes,
How did you set the parameters for the stimulation for each and every patient in your study for the stimulation?
Means parameters means how
The
much
strength of the stimulation, like
how much amount, madam?
the
How much?
current, or
Yeah, madam.
the
Actually,
current amplitude.
yes, amplitude and all right. Actually, madam, in a healthy person, if we want to use that device, we can use maximum level 4, madam.
Okay.
We have done that pilot study in the healthy people. 4 is the maximum level. And whenever this Parkinson patient starts, when he will connect the device with the app, that time the device they can set up, the level according to their comfort. Suppose they'll start with 1, they'll see how exactly they are feeling. 2, 3, 4, 5. If 5 they will start feeling dizziness, then they should come back to 4. So like that they can adjust and they can use this.
Okay. And have you tested the how long is this effect being lasted? Like how long? What was the interval of your follow-up study? Very curious.
Madam, yes, madam. 6 weeks and 12 weeks, madam.
Okay.
After 12 weeks, 6 weeks and 12 weeks. But the thing is that after 12 weeks also they are so interested, they wanted to use the device.
Okay.
And the problem is we have limited number of the devices. Because this device was given from Neurovalence UK, madam, for the research purpose. So we have limited number of the devices.
Okay. We have one more question from Tushar. In addition to verbal memory, if the motor balance was affected by stimulation, so they're interested to know if there was any effect on motor balance because of the stimulation.
Yes, madam. We have checked with the UPDRS scale and Berg balance scale. Postural stability was improved, and range of motion, that is mainly by physiotherapy parameters. So motor parameters also we have seen there is improvement in motor parameters also.
Okay. We have like 1 more minute, and there are 2 more questions. I'll try to quickly go through. How about habits like alcohol consumption and smoking? Did you look at screening those habits while you included the participants, and how was their effect on cognitive decline in the PD patients that you looked at?
No, madam, no, we didn't look it at, because getting PD patients is itself is very tough, and having very strict criteria, again, we may not get any samples.
Okay, okay,
Because as you have seen, we screened 169 people and got 34.
okay, okay. Um, okay. One last question, just very quickly. Have you checked the study effects against other methods like TDCS, TMS, TACS, like in terms of, have you compared literature?
No, madam. No, madam. In the human being, this is the first study what we are doing. I have already conducted in animal studies. Animal studies, I got a wonderful result. So this is my first human study as a pilot study,
Okay.
where I compared actual intervention with placebo.
Okay.
Definitely, I will take this message, and in future, definitely, I will plan to conduct a study.
Okay. Okay. Thanks a lot, Dr. Sai, and thanks a lot for the audience for all these interesting questions.
Thank you,
Let's
madam.
go to the next presentation. That's by Miss Nikki Singh from University of Kolkata. She's going to present. The presentation is going to be on the graded rise of inflammatory markers in the hippocampus induced by cerebroventricular streptozotocin (STZ) injection at increasing time points showed correlations with some memory impairments in rats. Over to you.
A very warm good morning to all. I would like to thank the organizers for permitting me to show my work here in this platform. My topic is the graded rise of inflammatory markers in hippocampus induced by intracerebroventricular streptozotocin injection at increasing time points showed correlation with some of the memory impairments in rats. As we all know, Alzheimer's is a progressive neurodegenerative disorder. The major neuropathological hallmarks of AD are senile flux and neurofibrillary tangles. AD is a heterogeneous disorder with both familial and sporadic forms. The familial form accounts for only 2 to 5% of the cases, but the great majority of the cases are sporadic in nature, with the possible risk factors as aging, type 2 diabetes mellitus, oxidative stress, and neuroinflammation. But very little is known about the exact mechanism of neurodegeneration in sporadic AD. So, in order to study that, several chemically induced rat models of AD are used in experimental research, such as ICV injection of colchicine, ocadaic acid, and amyloid beta peptide. ICV administration of streptozotocin is well established, validated, and widely accepted animal model to study sporadic AD. ICV injection of streptozotocin in rat, it induces neuroinflammation and dementia, which is similar to Alzheimer's disease and is used as a rodent model of sporadic AD. Most of the investigators previously they have reported increased neuroinflammatory markers in brain and memory impairments after 2 to 4 weeks of ICV-STZ injection in rats. But the exact relation between the neuroinflammation and memory impairments in ICV-STZ AD rat is not clearly understood. Hence, if the level of the inflammatory markers in brain or hippocampus are measured with gradual changes of memory impairments at different time points after ICV-STZ injection, the exact role of neuroinflammatory markers or the memory impairments may be investigated. Hence, our present study, it investigates the relation between inflammatory markers in hippocampus and memory impairments in ICV-STZ AD rats at different time points, like the 3rd hour, 24 hours, 7-day, and 21st day after ICV-STZ injection. Our specific objectives are to study the working memory error, reference memory error, latency to enter the first baited arm, latency to enter the 4th baited arm in radial arm maze, along with the level of CUX2, PGE2, TNF alpha, nitrite, and I0 in hippocampus at 3rd hour, 24 hours, 7-day, and 21st day after ICV-STZ injection in rats. And these results were compared with sham operated and the control rats. This is my study design. Rats were first habituated, trained, and tested for memory parameters in radial arm maze. Then they were divided as control group, sham operated group, experimental AD group. The experimental AD group, it received ICV injection of 3 mg per kg body weight of streptozotocin in 10 microliter artificial CSF at different time duration. The sham operated rat, it received ICV injection of 10 microliter of artificial CSF. Then further, the memory parameters and the hippocampal inflammatory markers were measured at the 3rd hour, 24 hours, 7 day, and 21st day post ICV injection. The following method and the materials we have used in our study. Now coming to the results section of the memory parameters. In case of reference memory error, we can see from the graph that in 24-hour study, 7-day study, and 21st day study, the errors increased in STZ-AD group as compared to the sham and the control group. And these changes were gradually increasing with time. But in case of 3rd-hour study, the memory parameters could not be measured as the rat, after entering the first weighted arm, it stopped the movement. Similarly, in case of working memory error, the errors were increasing. The errors in STZ-AD rat increased in 24-hour study, 7-day study, and 21st day study as compared to the sham group and the control group, and the changes were gradually increasing with time. Similarly, in latent sprint of the first weighted arm, the changes and the errors were increased in 24-hour study, 7-day study, and 21st day study in STZ-AD group as compared to the sham and the control group. And the changes were gradually increasing with time. But in 21st day study, after the 18-day, there were no further increase in the latency in STZ-AD group. In case of latency to enter the 4 weighted arm, similarly in 24 hours, 7-day, and 21st day studies, the errors were increased in STZ-AD group as compared to the sham and the control group. And the changes were gradually increasing with time. Hence, we can see from the results that the WME, RME, L1B, and L4B were increased at 24 hours, 7-day, and 21st day after ICV season injection compared to the control and the sham operated rats. The WME and the RME were progressively elevated at different time points in ICV STZ-AD rats, and the increased time in L1B and L4B in ICV STZ-AD rats were not gradually increased at different time points like that of WME and RME. WME, RME, and L4B were increased progressively from day 6 to 21st day after ICV STZ injection 21-day study. But the rat stopped movement at the 3rd hour study after ICV STZ injection, and the memory parameters could not be measured. But the sham operated rat, it showed pre-injection values of WME and RME at all the time points, including the 3rd hour after injection, which were quite similar to the control rats. The inflammatory markers, we can see from the graph that the hippocampal level of CUX2, PGE2, TNF alpha, nitrite, and INOS level increased in STZ-AD group as compared to the control and the sham group, and these were gradually increasing with time. Now, from the result of the correlation, we can say that the time-dependent changes of WME and RME were correlated with the graded increased level of the observed hippocampal inflammatory markers, but L1B and L4B were not correlated with these markers. Coming to the discussion, the mechanism of memory impairments in ICV rat and mouse model has been previously reported by several other researchers. The ICV injected rat develops insulin-resistant brain state, which is associated with the memory impairments, glucose hypometabolism, oxidative stress, and neurodegeneration that share certain characteristic features with the sporadic area in humans. The glucose hypometabolism in the brain may cause pro-amyloidogenic effect and the oxidative stress. The free radicals which are generated in this oxidative stress in turn causes DNA damage, mitochondrial abnormalities, and other changes that further lead to dementia. STZ also causes microglial activation, and which leads to the increased production of TNF alpha and IL-1 beta. From the study, the initial rise of the inflammatory markers after the 3rd hour of STZ injection probably affects the neurons and the circuitry of the brain that is involved with the movement and the memory retrieval. But this immediate effect was not probably due to an injection shock, as the sham-operated rats were not affected in a similar way. This immediate effect of the streptozotocin on the brain were changed with time, and the movements and the memory functions were gradually regained. All the observed memory parameters were affected in 24 hours, 7-day, and 21st day study due to neuroinflammation. WME and RME increased with time. The progressive increase of the inflammatory markers induced by streptozotocin is affecting WME and RME with a greater magnitude. The neural circuitries which is involved in memory retrieval are probably affected by these neuroinflammatory markers. The statistical correlation between WME and RME and the observed inflammatory markers probably indicates there is a strong coupling between the inflammatory markers and the WMNRB. The L1B and L4B are also affected by the inflammatory markers, but the correlation is not very strong between these parameters. Yes, we can conclude from the present study that the increased level of neuroinflammatory markers at the third hour after STZ injection is showed profound effect on RAS behavior, including impairments of memory and movement. All the observed memory parameters were affected by the inflammatory markers, but WME and RME appeared to be strongly coupled with the hippocampal level of inflammatory markers. Probably the neural circuitries for WME and RME through hippocampus were increasingly affected with the higher level of inflammatory markers. The present study has been funded by DFCC, SRI, Government of India. I would like to thank and express my sincere gratitude to the supervisor, Professor Tushar Kanti Ghosh, and the co-supervisor, Professor Debashish Bandyopadhyay, Department of Physiology, University of Calcutta. I'm also grateful to Ms. Ruksha Ghosh, Pritha Gupta, and other research fellow of Neurophysiology Laboratory. Thank you.
That's great. Thanks a lot, Ms. Nikki Singh. Okay. Okay. Sorry if I missed it, Ms. Singh. Is it what are the future directions that you're planning to take this work forward?
So here, what actually we have done is like we are seeing whether this neuroinflammatory markers they are affecting this memory parameters. So if we can, like in future, if we can just if we can block specifically these inflammatory markers, it might help in reducing the memory impairments that we are seeing in AD.
Okay, good. Thank you.
Thank you.
Let's move on to the next presentation. We have Mr. Varun Madan Mohan from NBRC. The presentation is going to be on a scale invariant perturbative approach to study information communication in dynamic brain networks. Over to you.
Greetings, everyone. Thanks for joining. My name is Varun, and I will be talking to you today about a simple perturbation-based method that I've been working on with Dr. Arpan Banerjee in the National Brain Research Center, which we've taken from a domain in network science and adapted to a neuroscientific setting. This method will help us gain some insights into the information flow in brain networks by looking at the influence of nodes and identifying nodes which are central to communication processes in the brain. Communication is everything in the brain, from scales starting from pairs of neurons to large-scale networks. Efficient communication underpins everything. And so it is also extremely important to know as much as we can about it. Communication is facilitated by the interaction of the dynamics with the network it evolves over. And as I'm sure you can appreciate, studying these network dynamical interactions over multiple scales is exceedingly difficult, which warrants the development of simple and easily translatable methods of investigation. That brings us to the heart of this work, which is using perturbations. The approach is very simply shown on the right, where the colored circles can be thought of as brain regions or even neurons, and the arrow shows that we have some activity or information flowing from green to red. Now, a perturbation is some exogenous change in the activity of green, which changes the information flowing to red. What we are interested in is these changes in activity, because how the activity changes is reflective of the intrinsic dynamics of the regions, and also on how these regions are connected with each other. The cited paper is the network science work which forms the basis of this study. So in order to test this method and understand what it can show us, we used an in silico approach. Also, to showcase the role of node dynamics in communication, we used two well-established neurodynamical models, which are the mean field model and the linear stochastic model. Right off, even to an untrained eye, these models look extremely different from each other. And we will show the communication implications of this soon. One thing I'd like you to look at here is this G, which is highlighted in red, which is a free parameter of the models. We arrive at the correct G by running these models, generating the functional connectivity and comparing it to the empirical functional connectivity. What's highlighted in green is the structural connectivity matrix, which for the whole brain modeling we get from DTI or DWI. We use the Euler numerical integration scheme, and since we are using these models simply to demonstrate the method as a test case, we set them to be noiseless. Perturbation protocol is very simple. We first initialize the dynamics on some network. That is, we set the C as the structural connectivity matrix that we want and let the system reach the steady state, which is like the resting state. Now we can see this on the left here in the diagram. We then choose one region, change its activity to a new value, as shown in the diagram, the green region. And what this does is the system now moves to a new steady state. As mentioned earlier, what I'm interested in is the change in activity between the original steady state and this new perturbed steady state. I put in these differences in activities neatly into a matrix called the response matrix. As an example, this is what the response matrix looks like for a network on the left running the mean field model. One thing you might have instantly noticed is how this matrix is asymmetric. That is, not everyone responds the same way when perturbed. So what do the rows and columns of the response matrix tell us? The column highlighted in green is populated with the responses in the network when node 1 is perturbed. That is the effect node 1 has on the network. The row highlighted in red contains the responses of node 1 when each of the other nodes in the network are perturbed. That is the effect the network has on node 1. Now, the difference between the green elements and the red elements actually gives us a very easily understandable metric of characterizing regions into those that are capable of influencing the network versus those that instead get influenced and follow these influences. We call this metric the net influence, which we define as simply the difference between the sum over rows and the sum over columns of R. A positive net influence denotes an influencer, and a negative influence would imply that a region is a follower. This brings us to this result. Here we applied the mean field model over an empirically derived structural connectivity matrix from the CAMCAN cohort for different values of the free parameter G, carried out the perturbations and computed the net influence of every region in the network. What we see is that there seems to be a very clear organization of the nodes in the network as influencers and followers, which depends on the free parameter G. At G star, which denotes the free parameter value that best fits the empirical data, we find an interesting influencer follower hierarchy across the core periphery axis, which is the high strength, low strength axis. What this implies is that if you want to effect the greatest impact on the network, you would want to target the high strength nodes, since they have the highest net influence. We see that this organization as influencers and followers is actually lost as we move away from this critical parameter value. So now that we know who the influencers and followers are in this network, and that you know that high impact nodes have to be targeted for an overall impact on the network, what exactly is the use of the follower nodes? Do they have any specific role? To understand this, we carry out the same perturbation protocol as before, but this time we freeze the activity of a selected node in the network. This functional lesion makes it blind to perturbations. The response matrix generated after freezing the activity of one of the nodes, say node 2 in this case, will not contain the contribution of node 2 in the transfer of any perturbation in the network. The difference between this functionally lesioned response matrix and the original response matrix thus captures the contribution of node 2 in all possible information transfer scenarios, or effectively its role in communication. This metric is termed as the flow, through node 2 in this case. Regions with high flow are central in the flow of information, like the blue node in the example here. You could liken them to gatekeepers. Even if I target the high influence nodes, the perturbations that they would transmit would not make as much of an impact if the high flow nodes are made blind to the perturbations. So the nodes you want to target to contain perturbation transfers are the nodes with the high flow. Now these perturbations could arise from neuropathologies, it could arise from some environmental factors, you name it. So these are the nodes that you would want to target to contain that flow or the transfer of perturbations. Here we've got the distribution of the flows of different nodes for the mean field model. We see that there is a clear intermediate strength dominance of flow at critical G for this particular case. This tells us that the nodes which control communication in this system, this particular system, are those with the intermediate strength and not those that are in the core or in the periphery. We also know that right before this critical regime, we have a very strongly core-dominated flow structure, and with high values of G, we get a very clear periphery-dominated flow structure. We also wanted to show the applicability of this method at a different scale, and so we computed flow at the level of the resting state networks as well. And here is what we see, is that when we use the mean field model, we find that the salience network has the highest flow when compared to the RSNs for this particular setting. So if you remember, I mentioned that we are interested in the change in activity brought about by perturbations because the amount of change would be dependent on the dynamics. This change is contained in the response matrix, which in turn decides the net influence and flow. So if the underlying dynamics changes, so will everything. This is very clearly seen here. On the right, we have the linear stochastic model on the same network as the MFM, and we see a very strong periphery-dominated flow structure. So, however, the physical implications of the metrics that what we've defined are clearly not dynamics dependent. What it means is that in this particular setting, if you wanted to contain the perturbations, you would target the periphery nodes. But in this setting, you would target these nodes here with the intermediate strength. Now here we see what happens when we compute the net influence and flow for a different data set. The NKI data set in this case, we see that the flow structure is qualitatively pretty similar over the data sets, which points once again to the major role of dynamics in how the activities change upon perturbation. In conclusion, we present here a very simple scale invariant method to look at network dynamical interactions. We show that using a construct called the response matrix, we can obtain insights into the organization of nodes in terms of their impact in the network, as well as on nodes that form the backbone of communication in the network, without a priori knowledge of the system's dynamics. In an experimental or clinical setting, you would use stimulations coupled with imaging to detect the changes caused, which would then populate the response matrix. A current limitation to this method is when we have a perturbation changing the qualitative dynamical behavior of the system, like a Hopf bifurcation. So the perturbations have to be controlled, and you need to know what it will do to the system. And finally, to reiterate the implications of the metrics, which I have to remind you is not dynamics dependent. This will always stay the same. If you want to impact the network, target the high net influence nodes. And if you want to control the spread of perturbations, target the high flow nodes. With that, I would like to conclude, and thank you for listening. I'll be happy to take any questions.
Thanks a lot, Mr. Varun. That's great. I'm curious to know how would you go about relating your simulation to say EEG data acquisition or any kind of imaging or any form of real brain data that you have in data sets, or you plan to collect it live, or what are your thoughts about just relating what you see now and what's happening?
Yeah, okay. Am I audible?
Yeah.
Yeah. So so a test case could be, say, suppose I want to do it on some fMRI study, something of that sort. So essentially I would couple this with some kind of a stimulation measure, like some way that I know a certain stimulation, say, I don't know, TAX or TDCS, something of that sort, something which I know would change a certain activity, a certain activation in some region. So what I would do is, initially what I would want to do is to get my response matrix. It tells me everything about the dynamics. So what I would do is I would give a very controlled sort of a perturbation and some kind of a stimulation to each different region that I'm interested in, right? And so the structural connectivity between those things can be given to me by MRI. And so after this, what I would essentially measure is how is my resting state changing? So a certain region which was not activated before is now being activated to a certain degree. And now the only requirement for me right now, as the work is done, is that the perturbation should be continuous. That's the only kind of requirement, even though even if it is for a small amount of time. If it is long enough to actually affect a certain change in the network, which is going to be stable with time. So if I have such a change, I can just measure these changes, which is very easily, you can do it for, especially for bold signals, even for any EEG or anything of that sort. And in this particular scenario with fMRI bold signals, what I would be looking at is the blood oxygenation level. So I would just measure the change in that, put it into my response matrix, and then do all these computations with that.
That's great. Okay, we have a question from Professor Sham. From a hypothesis angle, will the sensitivity of this node-based perturbation analysis be similar in dysfunctional networks?
I didn't quite catch that. Could you repeat that, please?
From a hypothesis angle,
Okay.
will the sensitivity of this node-based perturbation analysis be similar in dysfunctional networks?
By
Like how
dysfunctional
does the sensitivity of all these nodes and perturbation analysis that you have performed, how do all these change in case of dysfunctions? I think that's what.
In a dysfunctional network. Yeah. Okay. So the source of change when you're looking at I'm guessing by dysfunctional you're meaning say a disordered state. I mean, disease state or something of that sort.
Yes.
Yeah. Okay. So in such a state, I mean, we could the source of difference from a healthy, as far as this particular work is concerned, we are concerned with two things, which is one is the dynamics, how the dynamics changes, and how the network changes. So if you have a certain dysfunction which actually affects the network, then one thing I would kind of say is that, I mean, as long as the dynamics is compensated in some sense, if the dynamics works in such a way that it actually compensates for this change in the structural network, underlying network, I wouldn't actually expect to find any difference in the flow or such things, because the flow and especially the response matrix, because the response matrix actually depends on the change, and the change is dependent on the dynamics. So as long as the dynamics is preserved, I wouldn't expect any kind of change. But certainly you have dysfunctions which will also affect the underlying dynamics. So in that case, certainly. Yeah.
Great. Thanks a lot, Varun.
Thank you.
Okay, let's move on to the next presentation. We have Dr. Kusum Tual from NBRC again, and the presentation is on aperiodic and periodic components of ongoing oscillatory brain dynamics, linked distinct functional aspects of cognition across the adult lifespan.
Hi everyone, thanks for joining. My name is Kusum and I recently started my PhD at Hebrew University. Today I will be presenting the study about how aperiodic and periodic components are linked with distinct aspects of cognition throughout lifespan, which I worked on during my master's at National Brain Research Center with Dr. Dipanjan Roy and Dr. Arpan Banerjee. So we know that communication across brain regions occur via distinct oscillatory frequency bands, but aperiodic 1/f activity coexists with it, which is commonly referred to as noise. But recent evidence shows that the aperiodic component is not just noise, rather it is more dynamic in nature and critically influence oscillatory signatures, which most of the previous studies have not sufficiently taken into consideration. In this study, we use different measures for periodic and aperiodic components to get distinct functional insights about healthy aging process and cognition. But first, why it is important to dissociate 1/f activity. This is how a typical power spectrum looks like, which contains a predominant 1/f component following the power law function. The power in the alpha band is considered to be the total power as shown here, but rather the true oscillatory power is above this 1/f activity, which is shown here as relative power. Why? Because if we take the same signal with just a broadband shift, you see that the total power seems to be decreasing, but the relative power remains the same. So if we don't consider 1/f activity, it can lead to misinterpretation of results, as in this example. Total alpha power seems to be decreasing, but actually there is no change in alpha power. So to dissociate these two components, we use a recently proposed model called FOOF model by Wojtek, in which the periodic component is modeled as Gaussian peaks, which is defined by this equation here, whereas the aperiodic component is modeled by this equation here. And we used central frequency, power, and bandwidth to characterize the periodic component, and slope and offset to characterize the aperiodic components for further analysis. And when you take the linear sum of these two components, it gives you the model power spectrum. So the first data set we used was MEG resting state data, which was obtained from CAMCAN repository for 280 participants, age ranging from 18 to 88 years. For categorical analysis, we divided the participants into 4 groups, where each group had 70 participants. And to get the normative pattern with aging, we did continuous analysis, where we divided the participants into bins of 5 years, and central value was taken to be the representative age for each bin. The second data set of visual short-term memory task was also obtained from CAMCAN repository. In each trial, participants were presented with 1, 2, 3, or 4 colored discs for 200 milliseconds. Following that, a blank screen was presented for 900 milliseconds to hold those colors in memory. Then one of the original locations was highlighted by a thick white border, and at the same time, a response color wheel was also presented. Participants had to report by touching as accurately as possible the remembered hue of the highlighted disk, and 4 behavioral measures were estimated: precision, reaction time, VSTM capacity, and mean uncertainty. Mean uncertainty, which is a measure of metacognitive awareness, where subjects indicated their uncertainty in their choice of color. And then lastly, we categorize the set sizes into low and high load condition. Let's see the results now. Each figure is for each VSTM measure shown here. For all the figures, x-axis is the age in years and y-axis is the VSTM measure. These figures shows the age-related cognitive decline in all the estimated measures for both high and low conditions, which you can see in blue and red in color. Figure D interestingly shows that the mean uncertainty decreases with age, meaning that the old adults were more confident about their wrong answers compared to young adults. Next, we looked at how 1 by f aperiodic slope changes with age. Figure A shows that the 1 by f slope increases with age, meaning it becomes more positive with age. And significant group differences can be seen also here. The increase in 1 by f activity is suggestive of increased desynchronized neural activity. Additionally, this increase follows a nonlinear relationship with age, which suggests that desynchronized neural activity is not necessarily constant with age. Second figure is a representation of 1 by f slope values in sensor space, and we observe that the older adult shows more homogeneous distribution of 1 by f slope values compared to young adults, which means that the increase in 1 by f slope is not specific to any sensors. We then assessed whether the VSTM performance was impacted by 1 by f slope or not. So in these figures, x-axis represents the 1 by f slope and y-axis represents the VSTM measure, and each VSTM measure is plotted here. As depicted by these figures, we can see that 1 by f slope can significantly predict all the behavioral measures, as it has been shown that 1 by f slope is actually also predictive of measures in different cognitive tasks. We further suggest that age-related increase in aperiodic 1 by f slope does not necessarily mediate any domain-specific processing. Rather, it affects domain general processing. Next, we looked at the oscillatory powers of distinct frequency bands after removing the 1 by f activity, and we observed that the alpha power decreases with age, whereas beta and theta power increases with age. In the sensor figure, you can see that for theta power in older adults, most of the regions have no or less theta power. For alpha power, the power was found to be significantly coming from posterior areas, whereas the increase in beta power was mostly localized to centroparietal regions, which is generally associated with movement-related activities. In peak frequency measure, we observe a significant decline in peak alpha frequency with age. This decrease was not found to be localized to any specific sensors. Rather, a global significant decrease was found. Whereas for beta central frequency, it was found to be non-linearly decreasing with age, which was more localized to central parietal regions. Similar results were also obtained for bandwidth, band ratio measures, and also for other frequency bands, which are not shown in this current presentation. Lastly, as we wanted to see if these oscillatory chains are actually mediating any specific measures in the VSTM task, we carried out regression analysis between VSTM measure and these oscillatory features. And we found that after regressing out the age, alpha power is related to precision. Reaction time was well predicted by alpha central frequency, and VSTM capacity was significantly predicted by theta power. So you can see that different oscillatory features mediate specific behavior in VSTM task. For instance, alpha power predicted the precision of the participants, and not any other feature of the alpha or any other frequency band would predict the precision of the participants. In conclusion, our study first characterized the normal pattern of periodic as well as aperiodic components throughout adult lifespan. The observed age-related increase in 1/f slope seems to impact distributed processes of cognition and links with all the behavioral measures in different cognitive domains, suggesting that it affects global information processing. In contrast, oscillatory features of different frequency bands relates more to local processing and selective behavioral measures in VSTM task. And lastly, we can say that 1/f aperiodic activity certainly is not background noise, and it plays very important role in mediating cognition. Thank you for your attention. Feel free to ask any questions you have.
That's nice. Thanks a lot, Dr. Kusum. Are you here? Okay, we have a question. Could you elaborate on the possible sources of 1/f noise?
Okay, so there is no exact answer to this, but it has been proposed that the 1/f reflects the excitation inhibition balance in the brain, which comes from different neurons, inhibitory and excitatory. And when there is a change in inhibition and excitation balance, you also observe a change in 1/f. But there is no definite answer. There can be other factors also, such as you see altered tissue properties are also there, which change with age, or self-organized criticality if you go at more fundamental level.
Okay, that's nice. One more question there's another question from the audience. Could you explain how the mean uncertainty in VSTM task was calculated?
Okay, that's a nice question. So subjects basically indicated their uncertainty by the length of the time they touch the wheel. So you see a response wheel, right? So the length of the time they touch the wheel, that was taken as a parameter. And as they held their finger down, the white confidence interval was spread around the selected point, indicating greater or less uncertainty about the selection.
Just one last question, Dr. Kusum. Is there a measure of how aperiodic were the oscillations that you found in the brain? Like, we know the cyclic periodic nature of oscillations, but when you say aperiodic, of course, there could be different extents of aperiodicity. What were the characteristics of the oscillations that you saw in the brain for calling it as aperiodic?
So yes, so 1/f activity, when I say that's aperiodic or arrhythmic activity, it means that when you take a time series and convert it into a frequency domain, so you see a significant peak, right? So that most of the people consider as a peak or an oscillation. And if you see an oscillation or a peak in an alpha man, so we say, okay, there is a rhythmic activity going on after a certain interval of time, that is 12 hertz. But when you take the same signal and take that signal and convert it into a log frequency and log power, you see a straight line coming, which you also see in different signals when you take the LFP recording also, and in nature also 1/f activity is present. So 1/f activity is characterized by this slope, and there is no temporal dynamic associated with it. So there is no, we cannot say that, okay, the power is increasing, we cannot take the average, but it follows a power law function, which says that the scale is invariant. There is no temporal dynamics associated to it. And one of the other method you can use to dissociate 1/f activity is also spectral course analysis, where they say that if you sample the signal at different time, then the 1/f periodic remains the same, whereas the oscillatory parts keep on changing. So this is one of the parameter how you decide, okay, this is aperiodic and this one is periodic.
Okay, that's great. Thanks a lot, Dr. Kusum, for patiently explaining. this.
Just to correct, I am not yet doctor. I'm just starting my PhD.
Oh, okay. Sorry, I've been given wrong information then. Sorry about that, Kusum. Yeah.
Yes.
Thank you.
Thank you.
Okay, we are moving on to our next presentation. It is from Dr. Abhishek from Postgraduate Institute of Medical Education and Research, Chandigarh. The presentation is going to be on neurocognitive dysfunction in patients with opioid dependence. Does it improve on buprenorphine-based agonist treatment?
Hello, I'm Abhishek Ghosh. I'm an associate professor in the Department of Psychiatry, PGI-MER, Chandigarh. I am here to present our initial results of an ongoing study on neurocognitive dysfunctions in patients with opioid dependence. Does it improve on buprenorphine-based agonist treatment? I thank Cognitive Science Research Initiative, Department of Science and Technology for funding this study. I and other co-authors do not have any competing interest. In systematic reviews and meta-analysis, it has been consistently observed that opioid use chronically has been associated with a wide range of cognitive dysfunctions. The largest effect has been seen on psychomotor speed and cognitive impulsivity, whereas small and medium effects were seen in memory, working memory, and executive function. There has also been a possible association with the duration of opioid use or the drug-free state, meaning thereby an individual who has been using opioid today, if he or she stops using it for a while, some of these neurocognitive parameters may actually recover. To answer this particular question, Rapalli and colleagues published a paper in 2011. They have compared 3 groups of population, patients who are on treatment with buprenorphine, another group treatment on methadone, and they have compared this group with healthy controls. This is a graphical representation of how did these 3 groups fare in terms of the attention function, which was measured in that study by letter number sequencing task. We could see 2 months after initiation of treatment, both the treatment group did much worse than the healthy control group. However, the buprenorphine group would recover very rapidly. And on the second assessment, which happened 12 to 17 months after the initiation of treatment, there was no significant difference in terms of the attention function between the healthy control and the buprenorphine group. Therefore, it could be an early indication of improvement of attention with buprenorphine, which is an evidence-based treatment for opioid dependence. However, Rapalli study has very small sample size, perhaps that was inadequately powered. They have tested only attention and memory, although we have already observed that there is a wide range of cognitive dysfunction which happened in patients with chronic opioid use. They did not adjust for age and premorbid cognitive reserve. Also, they did not assess cognitive function on treatment-naive subjects. Why do we need to study cognitive function in general? Because cognitive function are associated with various functional outcomes like driving, operating machineries, doing executive work, ability to take important decisions in life, and problem-solving abilities. Given the importance of this subject area and caveat in the previous research, we thought that we do a 6 months prospective study of patients with opioid dependence who has started on buprenorphine naloxone to determine between group differences in cognitive functions of opioid dependence and healthy controls, and within group difference of change of cognitive functions of patients who are started on buprenorphine after 6 months of treatment, and between group difference between those who are on treatment and healthy controls. This is basically a pictorial representation of what I have already explained. This is a group of opioid dependence population who are tested for various cognitive functions at the baseline, followed up for 6 months, tested again. And these are the healthy control who are tested only once. And their cognitive functions were compared within group and between group comparison. We have 50 healthy controls and 63 subjects who are dependent on opioids. We did a wide range of neurocognitive testing for global intelligence, Wisconsin card sorting test for working memory, executive function, set shifting, Iowa gambling test for decision making, trail making A and B test for attention and psychomotor speed, visual and verbal NVAC tests for memory. Our subjects were in their early 30s. Most of them were men. The mean years of education in the opioid dependent group was nearly 11 years, in control was like 13 years. They were using opioids at least nearly for 8 years, and most of them were using heroin as their opioid of choice. The age did not differ between the opioid dependent group and the healthy control group. However, the healthy control group has significantly higher years of education. We have control for both age and years of education during our analysis. Now, what did we find when we compared the opioid dependent group versus the controls? As was expected, the control group performed significantly better in global intelligence. In various parameters of Wisconsin card sorting test, here I have only displayed one graph, that is conceptual rebel response, which denotes abstract thinking. IGT, the results are interesting, that's why I displayed it. You can see both the opioid dependent group and the control group have lost money. However, the opioid dependent group have lost more money than the control group. Now, when I say they were significantly different, these differences were adjusted for multiple comparisons as well. So our findings are robust. Again, these are the other parameters in which both this group differed significantly. Overall, when we plotted the effect size, we found that the maximum effect size was for the global intelligence, which was measured by standard progressive matrices. However, the effect size was 0.25, so it was a small effect. What did we find when we compare the treatment naive subjects, that those basically the baseline assessment, and 6 months after initiation of treatment? We observed a significant improvement in some of these parameters, like WCST total correct response. WCST total error was significantly low in the follow-up. That means the follow-up group performed better than the treatment naive group. Non-perseverative error, which is an indication of set shifting ability. Follow-up group again performed better. This conceptual level response or abstract thinking, follow-up group performed better. You can see than these individuals before they were started on treatment. Trail making A, again better performance of the follow-up group because they took lesser time to complete it. Verbal working memory, the lesser error score means follow-up group performed better. However, again, when we plotted the effect size hovered between 0.2 to 0.3. So it was a kind of very small effect. Now, this is where the results get a bit interesting. This is on treatment groups. So those who are on buprenorphine at least for 6 months versus healthy controls. You can see in the WCST conceptual level response, trail making B, and visual working memory omission error, the follow-up group or those who are on treatment perform better than the control group. However, in verbal working memory 2-back omission, which was a working memory test, verbal working memory test, here the control group performed better because they committed less errors. However, overall, the effect size remained small. So how to interpret this result? Patients with opioid dependence in our study showed a wide range of neurocognitive impairment, which had also been observed in previous studies. And more importantly, these impairments are not controlled, are not explained by age and cognitive reserves, which was measured in our study as a proxy years of education. 6 months after buprenorphine treatment, we observed an improvement in executive function, attention, psychomotor speed, and verbal working memory. Not all cognitive parameters, but some of the cognitive parameters have improved after 6 months of treatment. And more interestingly, the on-treatment group performed better than the healthy control in executive function, visual working memory, psychomotor speed, and attention. This was a surprising finding for us. What stood out in our result? Number one is the small effect size of difference between the patient and healthy control. As you would remember, the previous studies had observed an effect size of 0.5 to 0.7. So we wondered why. One of the reasons could be poor performance by the control. Why could the control perform poor? One of the reasons could be they could not, we need to develop tests which are indigenous in nature. As I have demonstrated once in the IUA gambling test, the control group had also lost money. They did not earn money, but they were supposed to earn money, the healthy people. So do we need need indigenous test, at least for decision making? That's kind of open question. We also observed a slow pace of improvement in treatment. That means these people will need a longer term treatment. One may also ask questions regarding whether all these neurocognitive parameters or these brain dysfunctions are reversible. Although we have limitations in terms of limited generalizability, and we don't know what happens after longer course of treatment. Thank you very much. I would love to take up more questions during the discussion.
Right. Thanks a lot, Dr. Abhishek. Any questions from the audience? Okay, I think probably one question from me. How have you looked at the gender effects also in your analysis, and how was that effect coming out in your interpretation?
Yeah, thank you for your question. Unfortunately, we have only one female patient in the opioid dependent group, and this was not by design. I would say this was by default, because most of the patients what we see and others see in various treatment clinics are men. So we did not adjust for the sex, but people who had adjusted for the sex in the previous studies from the West, they did not find any significant difference even after the sex adjustment. Yeah.
Okay, great. Thank you so much. Great presentation.
Thank you very much.
Thanks. Let's move on to our next presentation. It is from Ms. Aditi Katpalia from National Institute of Advanced Studies, Bangalore. The presentation is on measuring the level of consciousness in the brain using network connectivity approaches. Over to you.
A very good morning to all. I will be talking on measuring consciousness using network connectivity approaches. I am a postdoctoral fellow with the Institute of Computer Science of the Czech Academy of Sciences in Prague, Czech Republic, and this work was done in collaboration with Dr. Nitin Nagraj, who is an associate professor at the Consciousness Studies Program, National Institute of Advanced Studies in Bangalore. So consciousness and its measurement. So if you think about it, each one of us is having a conscious experience at the moment, and in very simplistic terms, this inner subjectivity of experience can be called as the experience of consciousness. So as humans, we have different levels of conscious experience. Humans can be in a coma state, anesthesia state, locked-in syndrome, then other states of consciousness like deep sleep, dream, waking state we go through almost every day, and then there is like samadhi state or hallucination state and so on. Measuring of consciousness, as we know, has very, very important clinical perspectives and applications, because first-person reports of consciousness are often not reliable or possible in the clinic. And for example, in the case of infants, they cannot report their state of consciousness, or in the case of locked-in syndrome, the person may not be able to report his experience. So lots of studies and theories of consciousness have been proposed since the 1980s, and however, we are interested in only some of the more recent work, which deals with not just giving a theory of consciousness, but also giving measures that can help to determine the level of consciousness. And The complexity theories of consciousness, such as integrated information theory, causal density, neural complexity, they allow us to do that. They allow us to measure consciousness, and what they are basically saying is that each conscious experience is both integrated and differentiated. So a conscious experience is experienced in one piece, it is perceived as a whole, and it is differentiated from any other possible conscious experience that we can think of or can be there. So because of this idea, they study the coexistence of functional integration and differentiation in networks to measure the level of consciousness in these networks. So these kind of studies are based on they look at information flow overlap or causal influence between different brain regions using different approaches to measure information flow or causal influence. And there are other studies which also apply connectivity approaches to specific brain regions, not the entire brain network. So now, since all these approaches are using the causal inference or information flow kind of measures to determine brain connectivity, we need to look at how robust these measures are, right? So in order to infer reliable connectivity. So correlation coefficient, for example, is non-directional, it is linear, and it is sensitive to the range of observation. So it has these limitations. Mutual information is again non-directional, it requires stationarity and estimation of probability distributions, and so gives poor performance on short length data. Granger causality again is a linear measure, although it is a measure of causality. It has several limitations, such as requirement of stationarity of the data, uncorrelated noise should be present in the data. It is sensitive to missing data and gives poor performance for short-length data, and so on. Transfer entropy again requires stationarity, estimation of probability distributions, and is sensitive to missing data, highly noisy data, and non-synchronous data sensitive. So we have recently proposed a measure called compression complexity causality, which helps to discover directional connections for given temporal data. And it is a method which employs complexity estimation techniques based on lossless data compression algorithms. So because of this, it is free of the assumptions of stationarity in the data or linearity in the data, or it does not require estimation of probability distributions, and is robust to short-length data, missing data, subsampling, filtering, and long-term memory in the processes. We tested for the performance of this measure on a number of data sets, and here we demonstrate a few examples where CCC outperforms some of the most widely used causality approaches like transfer entropy and Granger causality. And for the case of autoregressive processes with long memory, we see that when coupling is increased, TE and GC show confounding, while CCC shows very good performance. Even for the case of non-uniform sampling for autoregressive processes, CCC estimates are really robust even for increasing levels of non-uniform sampling or missing data in the time series. However, transfer entropy, Granger causality, non-linear Granger causality, they all fail even as the missing data points are increased to 5% of the original length of time series. We also tested Granger causality, which is the most widely used measure for some networks where there was some asynchrony. So one of the channels, we can say, gave data which was ahead of time. So even with like one sample asynchrony, we saw that extra connections were being discovered in the network which were not there actually. And also with two sample asynchrony, we discovered extra connections and one of the connections went missing. And as the sample asynchrony increased, some new connections kept coming up, some of the connections went missing. So, I mean, basically the point is that because of the problems with these connectivity measures and the fact that they are not robust even to small contamination in the data, everything can be screwed up in our information. So moving on, we simulated data from some large networks with 100 nodes, and these nodes in these networks were basically generating nonlinear chaotic time series of logistic maps. Nonlinear and chaotic data is also more commonly observed in the brain. So we took the case of no connections, the linear topology, and that of complete graph, where everything was connected to everything else bidirectionally. And what we observed was that the distributions of our bivariate CCC values were Gaussian, right? And for the case of no connections or only a few connections, the CCC was the mean of the CCC distribution was close to baseline CCC, and we observed low negative magnitude of CCC for the complete graph. We also simulated Watt-Strogatz logistic networks with 3 topologies: ring, which was the most regular kind of network, and the small world network, which was somewhere in between, and the random network. So small world networks are known to be most optimally sort of functionally integrated and segregated, and many studies have studied small worldness property in brain networks of healthy and undiseased people. So when we plotted the bivariate histograms for these 3 cases, we observed that in each case like the histograms were like bimodal. So the right distribution was probably reflecting the CCC values of the links which did not exist, and the left one of the links which existed. So what was observed was that the left distribution became more wider as the network became more random. So small world had a standard CCC distribution had a standard deviation somewhere in between the ring and the random topology. So there was something in the bivariate CCC histograms which was reflecting the degree of small worldness in the networks. And we also applied bivariate CCC to distinguish awake and anesthesia state in macaques. So what we did was we took recordings from 4 different monkeys, and these were acquired in a study by Yanagawa et al. And these were electrocorticographic recordings, and we took 3 windows of 5-second length each for both awake and anesthesia state in these monkeys. And anesthesia state was induced by this ketamine and medetomidine drug. So what we very surprisingly observed in the case of the histograms of awake and anesthesia state was that the awake state, just like a small world network case, showed more like a bimodal distribution of CCC values as compared to the anesthesia state, which looked more like a normal distribution. With the slightly long right tail, with showing few significant connections. And also the mean of the CCC values in the awake state was found to be higher than that in the anesthesia state across all the 4 monkeys, and the standard deviation was definitely consistently much higher in the awake state as compared to in the anesthesia state. So these were some interesting observations which sort of reflected what we had observed in simulated data. So with that, I end and would like to conclude by saying that CCC is a measure which makes minimal assumptions on the data and gives robust performance for challenging data, and functional connectivity estimated using CCC is distinct for networks which have high functional integration and segregation, as is supposed to be the case for highly conscious networks. And preliminary analysis shows that CCC distributions can help to distinguish between different conscious states. We have observed the performance. In future work, we would like to observe the performance of bivariate CCC on other complex network simulations, and compare with other existing approaches. And we would also, going forward, the aim would be to develop like a consciousness meter, which would help to sort of measure the level of consciousness. I would like to acknowledge the support of our funding agencies. I would like to thank my collaborator, Dr. Nitin Nagraj, and For your information, the CCC toolbox is available online, both MATLAB and Python implementations. Thank you for listening.
Very interesting. Thanks a lot for such a nice presentation. Any questions from audience? Okay, I think we have a minute or so for a question. I was curious to know, Dr. Aditi, about the significance of bivariateness in the distributions that you were presenting. Like what, I was just trying to understand what does that mean to have bivariate CCC?
Right, yeah, that's
Sorry
a
if I
very,
missed it.
no, it's a very valid question, because the thing is that when we look at connectivity, like we have multiple variables in the brain network, right? And we want to study how information is flowing between the recordings, like the temporal observations we are getting from each node. So we study causal inference between the temporal data obtained from like all, let's say, the electrodes that we have. What happens is that when we have like so many connections, it is possible that two nodes are not directly connected to each other, right? And there could be indirect flow of information from one node to the other. So very often people actually want to use what are called as like conditional causality estimation methods, so as to condition out the effect of other variables in the network, right? But the issue with CCC is that although it is so robust, we have still not come up with like a conditional estimator for CCC as of now, although we are still working on it, and there is some like we have done some work to extend it to at least like let's say 5-dimensional systems. But we still do not have a method which can encounter, let's say, hundreds of nodes. So that is why we estimated bivariate CCC, which means just between every two nodes. So we are like between every two nodes separately, and we are not conditioning out the effect. And the hope was that it will at least show out some emergent properties which can help to say something about like what happens when the network connectivity changes in various ways.
That's nice. All the best. Okay, just to save time, let's move on to our next presentation. We have our next presentation from Mr. Sandeep from Amrita Mind Brain Center, Kerala. And the presentation is on decoding hand grasp movement with EEG, characterizing gamma oscillations, temporal biomarkers, and other cortical potentials. Over to you.
Good morning, my name is Sandeep, PhD student at Amrita Mind Brain Center. Here I would like to present my work regarding how we could decode the grasp movement from EEG devices and the use of the gamma oscillations and other frequency rhythms. Our main objective of the work is to understand the population behavior of neural activity for movement with behavior. As we know, the movement statistics 2011 is shown as 2% with the disability population, in which 20% is the population is with movement disability. The rehabilitation for disabilities can be done by using BCA as an application if we can understand and decode the population activity. So what we know from the past, from 1970s, is that Rochelle and colleagues have shown decrease in rhythmic activity in alpha band power during ERP as indicating desynchronization. Also, they have investigated ERP cortical modifications with a delayed response in Parkinson's disease patients compared with healthy subjects over the contralateral sensory motor areas. Apart from these, MRCPs have been used as a biomarkers of movement intention and movement task in ALS, stroke, and Parkinson's disease patients. And from 1977 to 2004, activity related to grasping was reported to occur in lateral parietal frontal circuit involving the AIP primarily and the dorsal and ventral regions of the premotor areas. In 2010, Grafton and his colleagues have shown the connections of cortical grasping network in non-human primates involving anterior interparietal area M1, F5, and V6A regions as a reaching and grasping circuits. Reaching and grasping involves interacting neural processes which covers both domains of perceptual and movement related decision making. First, the surrounding environment needs to be perceived and sensory information is processed using the perceptual decision making. Second, there must be a preparation and planning for the actual movement, namely the movement onset and other movement details. In this work, we started EEG data acquisition using a 32 electrode device from 30 subjects, of which 23 males and 7 males, with a total of 173 trials. We have asked subjects to follow the cues in the presentation, to relax for 10 seconds. The plus sign indicates subject to alert for the next following task. As each word cue indicates the subject to reach the bottle which is placed in front of the table. And the next cue is a bottle image cue which indicates the subject to grasp the bottle, and the direction cue followed by 5 seconds is asking the subject to move the bottle to left or right directions, and followed by a blank blank cue or a blank slide where the subject has to get relaxed after the task. And all the cues are presented for 5 seconds each. After the data acquisition, the data was pre-processed above the 1 Hz with minimizing the drifts, and then band pass filtered from 1.1 Hz to 60 Hz with filter order of 4. And we removed the line noise using a notch filter, and we obtained some features of spectral and temporal domain using movement related cortical potentials and different frequency ranges. So to localize the electrode pairs or electrode regions for the movement task and relaxed task and alert task conditions, we have correlated among the different electrode pairs, and we found a relax and movement are quite indifferent, and movement and alert are having a higher correlation for central regions. So we tested a hypothesis test or t-test for relax and movement, which is very significant for relax and movement, whereas at alert and movement it is not significant. And among the regions for pre-movement and movement, we find a contingent negative variation over the before 500 milliseconds of the date movement. So we think that this could be a potential distinguishing marker for the movement and pre-movement, which from frequency domain perspective view, we quantified the oscillations of beta, gamma and alpha in both in central and parietal and frontal regions, and we observed a 29% decrease prominent during the relaxed task compared to the grasp movement task across the central regions. There is a 16% decrease was observed during grasp movement to post movement for the mu oscillations. And there was a 26% decrease for the mu oscillations from initiation to grasp movement. So these, apart from these, the major indications of features are 14% of beta and 10% of gamma oscillations. So these oscillations are shown a decrease from movement initiation to grasp movement, and there is also a rebound increment of these oscillations towards to the 14% and 20%. These indicates that these are the clear indicating features of which are distinguishing the movement, which we think they are distinguishing of movement. So later we use these features along with MRCPs to classify a pre-movement task and a movement task across from the central regions as the features, and we observed a 70% of training accuracy and 50% of test accuracy for pre-movement and movement. And also with this left hand and right hand directions movement, we observed a 76% of training accuracy, whereas 83% for the test accuracy using SVM algorithms mostly. And then we tried to decrease the number of features. We completely used the 74 features, both MRCPs and frequency oscillations, and we try to decrease the number of features by pruning the dataset using decision trees, but we have not attained a good accuracy rate over here. So in summary, we have seen a combination of readiness peak potentials, MRCPs, and mu, beta and gamma rhythms could be differentiating for pre-movement and a grasp movement. And we have seen a negative deflection at minus 500 milliseconds before the pre-movement, which could be a biomarker for the movement initiation and a movement task differentiation. And there is a rebound of beta and gamma band can be used for task-based evaluations for a grasp movement task. And these are all assumptions based upon the data, and the nonlinear SVM with the polynomial kernel performed both better than the some other classifiers like multilayer perceptron or decision trees. And in conclusion, we can say that the study can be done by recruiting more subjects. It can be extended by recruiting more subjects and increasing the number of trials. Expanding the study to explore microstates and frequencies of different frequencies could help to emphasize the greater role of surface EEG for preclinical assessments. Narrowing down the cortical regions of limb movements through EEG can allow low-cost EEG devices to explore the preclinical settings in rural areas of underdeveloped and developing nations, so that the telemedicine units can be empowered with the classification mechanism where it can be appropriate model. We can create appropriate models for diagnosing the healthy and diseased patients.
And this work supported and funded by grants of CSRI from Department of Science and Technology, Government of India. Thank you for your patience.
That's great. Thanks a lot, Sandeep. Nice work. We have a question.
Thank you.
What were the features that were input to the machine learning model? Was it based on different scalp electrode sites and regions? Was it like frontal, motor? I think you said motor, right?
Yes, we took from the features from the central electrode regions based upon MRCPs and beta and gamma oscillations.
From the central electrodes?
Yes, central electrode regions.
Okay. Sorry, was there any glitch in the presentation that you wanted to add, or was everything covered?
Yes, it is covered. It is covered.
Okay. Okay. One more question before we can just move on. Was there any kind of laterality effects that you saw because it was related to movement? And how about age effects? Like, was there anything on age too?
On the age kind of thing, we have a sample size of 7 females. So we can't, we have seen a significant difference between whatever the samples we have, but we can't come to a conclusion that there are significantly different based upon the sample size. And laterality, yes, we have seen some kind of laterality with the same sample size of 7 in the females, but not, as I said, there's less number of samples. We are trying to include the more number of female size samples, so we can come to some conclusion with that.
Okay, very curious. I think just few seconds. One last question. How about just training and testing on the same subject data versus training and testing with the population as a pool? Was there any difference? Have you tried it, and was there any?
No, ma'am. With the different subjects, I have not tried. We have take the data and completely on generalized with training and testing we have done.
Okay, with all subjects as training,
yes,
and,
yes,
and partition
yes,
that testing. You stratify
yes,
it and all that. Okay.
Yes, ma'am.
Okay. Okay. Thank you so much. Let's move on to the next presentation. It's from Ms. Priyanka Ghosh from NBRC, Haryana. The presentation is on distinct roles of MMN and P300 in processing prediction errors across modalities. Over to you.
A very good morning to everyone. Today, I'll be presenting our study, which talks about the distinct roles of MMN and P300 in processing prediction errors across various modalities. Since we very recently lost one of the greatest Kathak artists of our country, I'd like to begin my presentation with this picture. Let's say when the audience attends to the footwork of a dancer, say during a Kathak recital, the brain generates an internal predictive model based on the repetitive pattern of tempo of the footwork, as well as simultaneously integrating it with the tempo of the vocal recital. A single miss in the step of the footwork will lead to violation of expectation in the ongoing pattern and result in what we call as prediction error. Prediction errors in the brain are indexed by two ERPs, MMN or mismatch negativity and P300, which are elicited upon violation of regularity in a pattern of repetitive stimulus. MMN, as the name suggests, appears as a negative deflection in the EEG time series at around 100 to 250 milliseconds post onset of a deviant stimulus or a prediction error. MMN appears pre-attentively and has been even seen to be elicited in unconscious states like coma. P300 is a positive deflection with latency between 250 to 500 milliseconds distributed across frontocentral scalp locations. Being a late component, P300 is regarded as a cognitive marker. Even though there have been a lot of studies which have looked into the role of P300 and MMN, not one single study has talked about the relationship between the two ERPs temporally and spatially, especially in the context of processing multisensory prediction errors. Previous studies have shown that multisensory contexts speed up the processing of early sensory modalities like the N100 as compared to the unisensory counterparts. As a follow-up question, we intend to ask that do multisensory benefits extend to later ERPs that represent prediction errors like MMN and P300? Also, we want to validate if there is a sensory cognitive dissociation in the source distribution of MMN and P300. In order to address these objectives, we designed an EEG experiment where we recorded 22 healthy human volunteers in the age group of 22 to 43 years while they performed an oddball paradigm. Our oddball paradigm consisted of three modalities: audio-only modality, visual-only, and audio-visual. The audio-visual modality was the combination of the first two modalities, where the audio and visual stimuli were presented simultaneously. All the three modalities had two kinds of stimuli: the standard stimuli, which was presented in a repetitive manner or a frequent manner, and an oddball stimuli, which had a rare occurrence. 14% oddballs were presented per modality. Rest of the stimuli were standard in nature. The EEG data that was acquired was pre-processed and was grand average across all the trials of participants. The standard trials were subtracted from the oddball trials in order to visualize the MMN and P300 peaks, as pointed out by the arrows. For audio only, visual only, and audio visual conditions, the topo plots in the picture mark the sensor distribution of the MMN and P300 peaks. The darkest red regions denote the highest activity during the oddball condition. In order to get a fair comparison of the latencies and amplitudes across the three modalities, we picked the top 5 electrodes in each condition and computed their temporal projection on the first principal component for the standard and oddball trials separately. In order to check whether the two conditions differed significantly from each other, that is, whether the oddball condition was significantly different from the standard condition for MMN and P300 windows, we computed a simple student's t-test. The gray shaded regions on the left-hand side denote the areas of significant or the windows of significant difference between the oddball and standard condition. As you can see on the left-hand side for the MMN, the audio-only condition has the slowest latency as compared to the visual and AV. Whereas on the right-hand side for P300, we see that the visual modality is the slowest. Interestingly, the audio-visual condition takes a leading front in both MMN and P300. But in order to kind of validate if the AV condition or the audio-visual condition was actually significantly faster than the unisensory modalities, we performed a repeated measures ANOVA across all the subjects, followed by a Bonferroni-corrected multiple comparisons. Here we see that Indeed, the audiovisual multisensory context was the fastest for MMN peak latency, followed by the visual modality, and the audio modality was the slowest. For P300 peak latency, the audio modality was the fastest, very closely followed by the AV modality. The P300 amplitudes, however, did not show any significant difference between across the three modalities. For the MMN peak amplitudes, the visual MMN showed the least difference in magnitude between the oddball and standard trials. Next, we were interested to see if the generators of P300 and MMN sources differed across the three modalities. The MMN sources were different for different modalities. However, the P300 sources were common across the three modalities, clustered across the frontocentral regions, additionally showing up an activation in the FEF region for the visual modality. The MMN sources for the visual modality were localized at the secondary visual cortex, the right fusiform gyrus, the right temporoparietal junction, the right fusiform gyrus, the right temporoparietal junction. For the audio-visual MMN, we found sources that were localized at the multisensory integration regions of MTG and STG, along with a few activations around the visual motor region. The auditory activation was, however, localized at the frontocentral region around the supplementary motor area. This possibly explains the slowest latency of the audio MMN, as the travel time taken for the bottom-up information to flow up the hierarchical brain was highest in this case, which can probably be attributed to the auditory stimulus that was used in our oddball paradigm, where a higher frequency tone had to be recognized from a train of lower frequency tones, which probably initiated a stronger call for attention as compared to the visual stimulus, where one had to differentiate between two shapes or two colors probably, which was resolved at the level of the sensory cortex itself, because of which the visual MMN latency was shortest or shorter and tagged along with AV. With this, I would like to conclude our study with 3 points. The mismatch negativity serves as an index of initiation of attention switch towards change from a repetitive pattern, but it is modality sensitive in nature and depends on the stimulus and the modality properties. P300, however, is modality independent. And thirdly, the multisensory prediction errors are temporarily facilitated by the fastest unisensory modality, even at later stages of prediction error processing. Thank you.
Thanks a lot, Priyanka. That was very interesting. We have a question from Dr. Krishna Mepuram. What was the reference electrodes for grand averaged ERP plots? Are you here, Priyanka?
Yes, I'm here.
Okay. Would you like to answer that question?
Sorry, can you please repeat the question?
Yes. What was the reference electrodes for grand averaged ERP plots that you showed?
Yeah, right. So the default reference electrode while we were recording the EEG was CZ, and we re-referenced it along M1 and M2.
Okay. And the age group of the population that you were looking at?
The age group is between 22 to 43 years.
Okay.
The mean is 25, so it's on the lower side.
Okay. Yeah. Just one more question. Would you like to comment on how age would have an effect on the interpretation you had on different modalities with MMN and P300? Like, I know MMN is being measured even in infants and kids and all of them, right? So would you like to share any thoughts?
True. That's a very good question, actually. So, yeah, I mean, just not age, I would say that the level of consciousness perhaps affects these two ERPs a lot, because MMN is something which is seen even in unconscious states, say while we are sleeping or even in coma. So for someone who's not conscious, I mean, as we say that with mindfulness practices and yoga, and perhaps that is better with age, people do have a greater consciousness. So I would say that MMN is something that we would anyway see in the presence or absence of consciousness, but it would be very interesting to see the effect of consciousness on P300 perhaps, because P300 is a late ERP component, and that is elicited upon conscious intervention, when we are consciously attending to a task. So I would expect the amplitudes and especially the latency of P300 to probably speeden up, or the amplitudes to, I mean, so like there are studies, say for clinical studies for schizophrenia and depression patients, where they see that they have an enhanced P300. For aging, I'm not sure what is the condition, but I mean, the more attentive you are, or more the oddball is not an oddball for you, or I mean, you somehow expect the stimulus properties to be there, or more aware you are in that sense, that will not lead to a very higher amplitude of P300. Basically, the more odd an oddball is, the more higher the P300 amplitude would be. So I think with aging and experience, all these will come together and deplete the P300 amplitude probably, if that makes sense.
Yes, thanks a lot, Priyanka. That was very elaborative answer. And okay, we are nearing our last presentation of the day. It is by Ms. Shefali Gupta from IIT Delhi. The presentation is on neural correlates of understanding object concept in human brain. Over to you.
Hello everyone. The title of my paper is Neural Correlates of Understanding Object Concept in Human Brain. Here's the overview, introduction, present state of art, methodology, results, conclusion, and references. Coming to the introduction, humans possess a remarkable skill of recognizing an object, which they can do with great accuracy and ease. This skill in itself raises one of the most important and fundamental question of neuroscience, which is how the visual system interprets the environment around it. In other words, how the brain recognizes or learns a particular object. Many researches are being carried out to understand this mechanism of brain. It is believed that an object is recognized by grouping elements it is constituted of. In the present work, we have attempted to provide a new insight to the existing theories of grouping perception for recognition of object in the brain. To understand grouping perception in brain, we have attempted to evaluate if there exists any difference in response to structures and non-structures. Coming to the present state of art, research has been carried out on how structure or contour is formed and how that is perceived visually. This concept is tried to understand through different modalities such as EEG, MEG, fMRI, MRI, DTI, etc. So, we have tried to explore the concept of perceptual grouping and provide a deep insight to it using EEG. Coming to the methodology, to understand how an object is recognized, I have considered some basic structures. So you can see here some basic structures. My objective was to see if the response to structure in brain is different from non-structures. Here you can see images containing random dots, out of which in the images in the left side, some dots are forming a line, a structure, which is nothing but the two lines either touching or crossing each other near the center of the image. while the images in the right side just contain some random dots. I have presented these images of different structures and non-structures to the participant while collecting the EEG data for about 1 second. So here is the experiment design. The gap between the image was maintained of 1.2 to 2.3 seconds. There were 20 images of both events repeated 5 times, thus 200 events, 100 of each structure and non-structure were obtained. That's how the experiment was designed, and the overview is shown in the figure. Coming to the apparatus, I have used for data acquisition is shown here. Firstly, the technique that I used here is EEG because it provides great temporal resolution, and thus the neuronal activity even in the range of milliseconds can be captured. As shown in the figure, the 64-channel brain vision recorder system was used, which is available in the neurocomputing lab of IIT Delhi. I had 2 screens here connected in sync with each other, out of which one is used for stimulus presentation while other was used for collecting the raw EEG data. The participant was prepared by placing the electrodes on scalp with an conducting gel. He was seated at a distance of about 40 to 60 centimeters from the screen and was instructed to avoid any body movement and focus on the screen. Proper participant consent was taken and proposed experiment was approved by IIT Ethical Clearance Committee. The data was collected for about 15 subjects in the age group of 19 to 30 years. None of them had any neurological deficit. Coming here are some glimpses of the subject preparation in the lab. Coming to the analysis, the pipeline of analysis is shown in the figure. As seen in the pipeline, the raw EEG data was firstly pre-processed to remove any noise present in the data. Then the electrode sites showing significant difference between the 2 events, ERP in frontal and occipital region were determined. The responses of those electrodes in frontal and occipital region were averaged. Similarly, ERP in frontal and occipital region were averaged for all subjects, and thus GERP was calculated. Also, the T-score analysis was performed to check the statistical significance. Coming to the results. Firstly, ERP results. I have calculated ERP for frontal and occipital electrodes for the events, structure, and the non-structure. It is found that the frontal region ERP incorporates 3 major ERP components, namely P200, FN400, and LPC. P200 is a positive peak around 150 to 275 milliseconds after stimulus onset and is responsible for stimulus classification. N400, also known as FN400, is a negative going wave that peak around 300 to 450 milliseconds post-stimulus during recognition task. FN400 indexes repetition-related process and indicates potential for conceptual integration. LPC, which is the late positive component, it generally elicited around 600 to 100 milliseconds after the stimulus onset and is a positive deflection and is related to more attention or feature by feature comparison of the stimuli. The resulting ERP is checked for significant amplitude difference between structure and non-structure for all its components, P200, FN400, and LPC using ANOVA analysis. Thus, the more activation for structures can be seen at the frontal region as compared to the non-structures. Coming to the occipital ERP, which is shown in the figure 4B, it is observed that ERP at occipital region incorporates 4 major ERP components, namely P100, N200, P300, and LPC. P100 wave refers to the positive deflection around 90 to 150 milliseconds after stimulus onset and is sensitive to visual cognitive processing. N200 wave is a negative wave that peaks around 180 milliseconds post-stimulus and is driven for highly unfamiliar stimuli or stimuli with complex object. P300 wave generally elicited 300 milliseconds after stimuli presentation and is a positive deflection and indicates discrimination of one event from another. The resulting occipital ERP was checked for significant amplitude difference between structures and non-structure event for all components. P100, N200, P300, and LPC using ANOVA analysis. One more thing you can see here is difference wave in black color in both the ERP figures. The point of divergence of difference between the events for the frontal region ERP occurs around 100 milliseconds, whereas for the occipital region, the potential for two events diverge sharply around 60 milliseconds after the stimuli onset. The point of divergence is an important EEG feature as it reflects the target detection by the brain. I have also calculated ERP for electrodes in primary visual cortex area V1. The ERP wave gives an additional component as compared to previous occipital region ERP, which is C100. It is believed that when information enters through visual sensory input, it is received by V1 area and C100 wave confirms the reception of that information. The difference wave here showed first significant negative peak around 60 milliseconds after stimulus onset. Thus, the neuronal processing and the biological markers related to grouping perception are evaluated and examined in frontal, occipital, and V1 areas of the brain. ERP results suggest the perception of grouping induce higher activation in the brain as compared to the other stimuli. Most importantly, the results suggest that the grouping perception can be perceived by the brain in merely 60 milliseconds. This is the most important result. Now coming to the GERP results, the ERP of all 15 subjects are averaged in frontal and occipital region to obtain GERP. GERP is the grand event related potential. The GERPs showed similar concept, similar components as shown by the ERPs. GERP also showed clear and statistical as well as visual difference between structure and non-structure. The difference curve for all 15 subjects in frontal and occipital region is also showed. Further, T-score for the regions is also shown separately. If the two events are statistically different, T-score will show high value. Low value of T-score will represent the T-value between the two similar events. Now coming to the next result, which is the flow of information. Flow of information can be judged by topographical maps presented in figure. These topo maps follow same result as ERPs, such that at 60 milliseconds, activity can be seen at occipital region, while for frontal region, activity can be seen at 100 milliseconds, which were the first point of potential divergence for the occipital and the frontal regions. Coming to the next point, which is the information flow from frontal to occipital region of the brain, which is indicated by the topo plot at 100 and 140 milliseconds. And similarly, the ERP results can be judged and were followed by this flow of information graph. Coming to the concluding, so I can claim that the ERP results suggested grouping perception induced higher activation in the brain as compared to the other stimuli. Also, the presence of C100 and also the first point of divergence for the occipital region that occur around 60 milliseconds, evident that the brain can perceive structures in just 60 milliseconds. Also, the presence of C100 wave indicated the flow of information from V1 area to the frontal area of the brain for structures, which signifies the feed-forward processing in brain for perceptual grouping. Here are some of the references. Thank you.
Thanks a lot, Ms. Shefali. That's great. Any questions from audience for our last presentation? Okay. Okay, let me end with one question then.
Yeah.
I was very curious, what made you get interested in this object concept per se, and where do you see this work going forward?
This is a very basic study or the study related to basic science. So as we are moving towards the machine learning models and other things, to train those models, we need to know the basic science behind it. So this is the very basic science which is really needed to make our models more effective. And these many such studies are going on, and a lot of literature is there. And actually, we are going to take these results and compare to the congenital subjects which are blind. So which is not presented here, which is my future objective. And that's how. Thank you.
Great. We have one from Dr. Krishna Mayapuram.
Yeah.
What was the key novelty of your research?
So the key novelty or the important result that I found here was that the brain can perceive structures, or that can group the objects in merely 60 seconds. So that was the most important result of my work. And
cool. And what is C100 and where is V1 electrode?
Okay, so V1 area is the primary visual cortex area. So in that image, I have shown marked the electrodes in the area, primary visual cortex area. And so for those electrodes, I have calculated the ERP, and thus I got the C100 marker, which shows that the information is being received by the visual cortex area, and it is there in the literature.
Okay, great. Thanks, Shefali. That was great. And thank you, everyone, each and every one of you, for your time and patience and keeping up with the hack. And thanks a lot to the presenters. All of them were very wonderful and exciting. I thank Arati for helping me co-chair this session very wonderfully, and all the team here. And thanks a lot for this opportunity. I truly enjoyed it. I'll leave it now to the organizers.
Dr. Pragati, Priyadarshini, it's been an amazing pleasure to have you chair the session, and we're very, very grateful for your time and for your effort and for being so kind with all our presenters as well. Thank you so much.
Thanks, Dr. Shyam. Thanks.
Thank you. Thank you so much, Dr. Pragati, for the very systematic handling of the session. And let me speak from my heart that your calm and humble-natured presence in our conference was such a pleasure to experience. Thank you so much, ma'am.
Thanks, Zakir.
Okay, and I would like to thank Mrs. Arati for the profound support that you gave for the smooth conduction of our session, and my sincere appreciations to all the speakers of the talk session. You guys made the sessions like filled with enthusiasm and valuable information.